welcome back for this session on continuation with the biomedical optics. So, before we move forward, let us understand what are different constituents of the tissue which is present within this biological organization. So, what you see over here is what do we have over here? We have hemoglobin within these tissues. Hemoglobin also has so what is the purpose of hemoglobin? The purpose of hemoglobin is to transport the oxygen. So, it has the ferrous compound inside or ferrous ion which actually gets attached to this oxygen, and it transports the oxygen from one place to another. Now, there are two states of hemoglobin, the one being the oxygenated hemoglobin and another is deoxygenated hemoglobin. So, in this case what you see is that there are two different ways to characterize the hemoglobin, that is oxygenated hemoglobin, that is also known as HBO, and deoxygenated hemoglobin, that is known as HBr or reduced hemoglobin. Another constituent is collagen, which drastically affects the optical technique. The third one is the lipids. One of them is these lipids that you see on the phospholipid that you see as a which is part of the basal membrane, and then finally is the next one is the water. So, in the case of water, what you see is H2O, and the oxygen makes a particular angle with the hydrogen ions. So, let us see what happens once we pass the illumination, that is the light, through the tissue. So, what do we see? We see that there is certain type of laser or light that I am passing. So, I am passing a pulse of light. I am not passing the continuous light, but I am only passing the light for a very small duration of time. And what you can see on the right is there are different types of photons, or not types of photon, the type of photon is only single, but the way that the photon interacts with the tissue is different. For example, what you see is the ballistic photons. So, this ballistic photon, which is in green color, is the one that reaches the first. Okay. So, here you can see that it reaches the first, that is the green color, and the next is the sky blue colored snake photons. So, the ballistic photon, as you can see, does not undergo or goes very minimal number of scattering events. Snake photons are the photons which undergo the scattering which is again not many, is like less number still, the lowest being in the case of ballistic photons. And then finally, we have the diffuse photons. The diffuse photons are the photons which scatter the highest. In the case of a turbid media such as tissue, we will always see the cases of diffused photons when the tissue dimension is like more than 1 mm or 2 mm. Okay. So, in these cases when the dimension of the tissue increases, in this case in such cases the diffusion nature is going to be more dominant. And in the next few classes we will also talk about the diffusion process, we will talk about the transport equations and how those can be used to characterize or reverse engineer the optical properties of the tissue, such as scattering coefficient and absorption coefficient. So, here you can see that the ballistic photons are the one which goes minimum scattering and arrives first, and then it is the snake photons over here which arrives after the ballistic photons, and then the whole lot of other photons that you see over here are the diffused photons that arrives after the snake photons. And you also see a certain trend over here that it is not actually a Gaussian, but it has this peak has a long tail, and this tail would also help you to understand the scattering coefficient of the tissue. Okay. So, we will get to know about this at a later stage once we try to learn the mathematical model, but this is what is actually happening in the hindsight of the light which is propagating through the tissue. So, if we want to now characterize how much scattering is going to happen or how much absorption is going to happen, we introduce two variables, the first being scattering coefficient and the second is absorption coefficient. So, in the first case that you see in the top, we assume that this top part of the tissue is highly scattering. In such cases you have mu underscore s, that is mu s is higher as compared to the absorption coefficient, that is mu a. So, in that case you can see that there is a lot of scattering, that is scattering events that are going to occur, and this is the current state of the photon for example here. Some of the photon you can also see that actually comes out of the tissue, which would not be actually getting detected by the detector which is placed on the right side. Okay, very few photons you see that are actually being detected. Some of the photons actually get reflected backwards. So, you can also have a detector on the back side to actually detect the backward reflected or backward scattered photons. In the bottom part, what we assume is that mu a, that is the absorption coefficient, is higher as compared to the scattering coefficient. So, what we observe over here is that the photons are getting absorbed rather than getting scattered. So, here is still getting scattered, as you can see over here there are slight scattering events, and but eventually they are getting absorbed within the tissue. Okay. So, in such cases you have absorption coefficient is more than the scattering coefficient, but in most of the cases of the breasts where there is a presence of tissue, you will see the event or on the top, that is the top case that you see over here, where the scattering coefficient is more than the absorption coefficient. For the other contents where you have more fluid content of the stroma of the breast tissues or the extracellular matrix, you will see the bottom kind of events or bottom kind of case which is going to be more dominant. So, this is the case where you see the macroscopic view of the interaction of light with the tissue, but what about microscopically? So, what if we draw the cell structure and then see how the cells are interacting with the light. So, let us draw a cell tissue over here. So, not a tissue, but only a cell over here. So, what you see over here, this is a single cell, it has many organelles on the right side. So, it has the Golgi apparatus, it has nucleus, this is the nuclei, okay, and the other components or organelles within these cells over here. You have the mitochondria, and this is the light that I am going to pass through this cell. So, what do we see over here? We see that some of the organelles are actually refracting the light. So, the light is passing through this and then it is getting refracted by a certain angle. We also see that the light is hitting the nucleus inside this cell and it is getting back scattered. Okay. Some of the photons which pass through the cells and it gets refracted to a very high angle, very large angles, and then again there are some other photons which are getting refracted again with some other organelles of the cells. So, this is what happens in microscopically once the light interacts with the tissue. So, this being just a cartoon, but eventually there are a lot of number of cells which are present and this occurs in lot of instances. So, let us go forward and come up with how the theory and mathematical modeling happens for the light tissue interaction. Now, coming to the first part is where people started to work with Beer-Lambert law. So, you have the Beer-Lambert law. So, what do we have over here? So, we have and this is the case where your assumption is mu a is more than mu s. So, your absorption coefficient is far higher than the scattering coefficients, and you take a tube for example and you pour a liquid over here and this is strictly for this kind of cases only, not in the case of turbid medium such as tissue. And what we do is we have this LED or the laser, and we shine the laser over here, and it undergoes some scattering, and then it comes out from over here, and here we have the camera. It could be a CCD, for example, charge coupled devices over here, and what we see over here is the light intensity, that is I naught, and the light which is getting detected by the detector. So, over here this will not be I naught, it would be I. So, now if I want to relate this I with I naught, that is initial and the final. So, how do I relate it? So, it is related by I by I naught, which is equal to exponential minus alpha L, and L is the thickness of your column or the tube that you are pouring a particular liquid, and alpha over here is the property which is exhibited by the liquid. So, as you can see over here, if you plot it, as the length of the tube actually increases, you will see an exponential decay of the optical density. So, it is also known as optical density, that is a log of the ratio, or you can also call it, it might not be the correct way to define the optical density, you can also call it as kind of a attenuation. Okay. So, how much the light is getting attenuated, it is in a walk sense, you can just tell it that it increases as the length gets increased, the attenuation is going to get reduced. In other words, your A is getting reduced when the I, that is the light which is traversing the liquid medium, is getting reduced. So, this is the case when we have the Beer-Lambert law, which is a very simple case when we have the liquid present in the column. But when we have the tissue, so for example, when we talk about the tissue, so light which passes the tissue is not going to just go like this, because it is having a lot of components, as you already see. So, you know, even a single cell has many components. So, it will not just go just straight out of this tissue, but it will undergo a lot of scattering over here, and some of it can go outside, some of them can actually go forward also, and some of can go backwards, some of them can go with a particular angle. So, it is going to be the absorption is not going to be more dominant, but the scattering is going to be more dominant. So, the Beer-Lambert law might not be the best way to actually quantify the optical properties of the tissue. So, which law is going to be better? So, what people did is they come up with a new set of laws for the optical propagation through the tissues, and that is known as the modified Beer-Lambert law. Okay. So, here you can see this, we talked about I by I naught earlier, this is the attenuation. If you take the log of that attenuation, you get this optical density, and that is again a function of a particular wavelength. So, again you have this tissue, you pass this light over here, so you have I naught and you have I. Okay. The light that you pass is also having a certain wavelength. Okay. So, if you see this lasers have very short bandwidth. So, for example, you have 650 nanometers wavelength. So, it has a very less bandwidth, maybe around 5 nanometers. But in the case of LEDs, and that's what our lab is working on using very cheap LEDs. You have very high bandwidth. Again, we still are working with 650 nanometers, for example, and but it has a large bandwidth over here, which is around 40 nanometers, for example. Okay. So we have the function which is of which function dependent optical density, which is optical density with the function of lambda, and then we have again the absorption coefficient. Okay. So this is again a function of wavelength, and then we have introduced a new term which is known as differential path length. So here is the definition of differential path length, okay, again which is a function of lambda, and then this is the d, that is the thickness of the tissue, okay, and this is d over here, and then finally we have the factor g. So this g is actually the attenuation factor, and that is from both the geometrical and the scattering coefficient, and we do not want this term. So, we will try to remove this term out of the equation, and the way to do that would be we are going to make a lot of assumptions. The first assumption is that the geometrical configuration for all these measurements is going to be the same, okay, and the scattering which is going to occur from them is also going to be the similar. Okay. So, that is a kind of assumption that we do in this Beer-Lambert law. So, we are going to use this Beer-Lambert law, and finally, we are going to quantify the optical properties of the tissue, mu A, mu S. We also have other properties of the tissue. Okay, so let me just so we quantify mu A, you quantify mu S, and we also have quantified the volume fraction of the different constituents of the tissue. As we talk about lipid, we talk about collagen, we talk about water, and then we also talk about hemoglobin. Okay. So, these are the different constituents that we can actually quantify, the volume fraction of those constituents. Okay. Looks good. So, we are going to use this modified Beer-Lambert law instead of the Beer-Lambert law. Okay. So, going to the next is how do we eliminate the G, that is the attenuation factor. Okay. So, this is the same equation, modified Beer-Lambert law. Earlier we only had mu A as a function of wavelength and d, but now we also have introduced the dpf and the g. Okay. So, in order to actually do away with the g over here, what do we do is we go to a differential modified Beer-Lambert law, which is also known as d MBBL. Okay. And with this we can actually do away with the g part, that is we negate the g part. So, what do we add is this delta term to negate the g part over here. Okay. So, now we do not have the mu A, but mu A is the difference in the mu A as a function of wavelength for two different states. The two different states could be like inhale and exhale would be a simple example, right. So, it could be some other way. For example, we were talking about analyzing the quenching of the hand tissues. Okay. So, quenching of the tissues could be one of the case, or when we inhale and exhale. So, when we inhale, you have more oxygen present inside the lungs, okay, and when we exhale there would be less oxygen which is present within the lungs. So, this also going to change the two different states of the properties. Okay, looks good. So, so we already have the absorption coefficients for different constituents such as lipid and collagen for different wavelengths. Okay, for 850 you can see that lipid has 0.637 per meter of mu a, that is the absorption coefficient. For 935 again we have the absorption coefficient for lipid and collagen, and for 1060 we also have the absorption coefficient for lipid and collagen. So, what we see is that at 850 nanometers you can see that the absorption coefficient of collagen is very high as compared to the lipid. Okay. In the case of 935 you can see that the absorption coefficient of the lipid is more as compared to the collagen. So, based on this we can actually quantify the constituents of the tissue or the composition of the constituents of the tissue. For example, how much is the constituents of the collagen and how much is the constituents for the lipid. So, for example, what we do is, again if I go to my screen over here, again we have the same tissue. So, instead of passing through a single light lambda 1 and detecting, so I will have I naught of lambda 1 and I detect it as I of, oh sorry, I need to make it lambda of lambda 1. I detect it by a detector with I of lambda 1, and I have one more light that I pass through this tissue. I have I naught of lambda 2 and I detect it I of lambda 2. Okay. Now, what do we have is, for example, we say that this tissue has different constituents. So, for example, it has lipids, it has collagen, it has water, and it has hemoglobin. As we discussed, hemoglobin could be of two states. It could be oxygenated hemoglobin or it could be reduced hemoglobin, which is also known as HBr. So, either it would be carrying the oxygen or it would be not carrying the oxygen. So, in this case oxygen is not there, but in this case the oxygen is present. So, in both the ways it will actually behave differently. Okay. For example, if you now draw the wavelength as a function of absorption or mu a, what you observe is that for the water, for example, the peak will actually rise somewhere at 975 nanometers, and then it reduces and then it finally increases after that wavelength. In the case of lipids, what you see is that there is a peak of absorption specifically at 940 nanometers. So, in the case of water it is around 975, in the case of lipids it is around 940, and then for the other constituents such as collagen, you can see that collagen has very high absorption and it is like similar to this. In the case of collagen, so over here it is like very high in the case around visible range of light, and that makes sense also because it is kind of a fibrous content and it actually absorbs a lot of light within this wavelength, maybe at 850 nanometers. Okay, we can just write it as 850 nanometers, and that is what we have used over here. Okay. So, the way the light actually gets absorbed at different wavelengths is different for different constituents. For hemoglobin again it varies accordingly. For both hemoglobin oxygenated and deoxygenated, it actually varies. Okay. So, what do we have over here? So, what we have is two different wavelengths, okay, lambda 1 and lambda 2, and we detect lambda 1 and lambda 2 light from here. Now, let us go to the our slideshow. Okay. So, this is what we are doing right now. We are passing the light through the tissue at different wavelengths. The number of wavelengths will depend on the number of constituents that we want to evaluate. Okay. So, for example, coming back over here, if I want to evaluate only lipid, collagen and oxygen, these three constituents, I will need to have 3 different wavelengths. Okay, I need to have lambda 1, I need to have lambda 2 and then finally lambda 3, and from the modified Beer-Lambert law I will get 3 equations. Okay. So, I have 3 unknown. So, this is my unknown 1, unknown 2, unknown 3, and I have 3 equations because of 3 different wavelengths. Okay. So, I am able to now calculate the composition of lipid, collagen and oxygen, water. If I want to have 4, I want to quantify 4 constituents, then I need to have 4 different optical wavelengths. Okay. In our case, in our lab, we did it with 2 optical wavelengths, that is lipid and collagen only. So, we had operated at 2 wavelengths. Okay. Okay, that's good. So, let us move forward. Over here, what do we observe is there are 2 different parameters. The first one is mu A and second one is dpf. In the case of differential path length, I need to make this a more simpler implications. So, what do I do is instead of assuming the light to be passing through this tissue like this and getting detected by like this, what I assume is a simple infinite slab model. So, this is on the right side, this is known as infinite slab model. So, which means that the source that you see over here is somewhere over here and this is the detector, and both the source and detector are inside the tissue, okay, completely within the tissue. I am not assuming it as the light is coming which is present outside the tissue, I am assuming it to be inside the tissue. It is kind of a very simplistic, the most simplest assumption that you can use to proceed further. With this approach, what I can do is I am going to simplify the dpf for the infinite media over here, and I take this as the derivative of the log of the intensity, and then I can write it in the form of the effective mu, that is the effective attenuation coefficient, and with this that would be a derivative with respect to the mu A, and finally what I do is I get this equation, okay, and where mu s prime is again related to the operating wavelength over here, and this A and B are the property of the tissue, A being the scattering coefficient and B being the scattering power. So, for different tissues or different parts of the tissue, for example, lipid, collagen, fat, adipose kind of tissues, fibroblasts, so all of them have different constant or different values of A and B, okay, and lambda naught over here is the reference wavelength. And that is just to make this as dimensionless, okay, makes sense. So, with this now we have simplified the dpf to mu A and mu s, and we are going to quantify the mu s over here with the equation of A times lambda by lambda naught to the whole power minus b. Okay. The next part is the mu a. Now, how do I simplify the mu a in the terms of tissue constituents? So, one of the way is to write the mu a into small contribution from each of the constituents. So, for example, what I am going to do is I am going to add one more page over here, and what I assume is the tissue over here, and this tissue, for example, has only two constituents. For example, I have lipids and collagen. Okay. So, what I assume is that the contribution from lipid would be mu a comma L, and contribution of collagen would be mu a comma C. Okay. So, what I can see that complete total absorption or total absorption coefficient would be the summation of volume fraction times the absorption coefficient for each of the constituents. Here I, which is going from 1 to 2, I assume only two constituents, or actually it can be n. Okay. I going from 1 to 2, it could be n, I going from 1 to n. Okay. And n, I am assuming it to be 2 over here. Okay. So, I can elaborate my equation to f v comma L into mu a comma L, okay, plus f v comma C into mu a comma C. Okay. So, this is my absorption coefficient for two different constituents. Okay. Now, I can also make it a function dependent mu. Okay. So, for example, this I am going to write it that mu a, okay, mu a is going to be for a certain wavelength lambda 1, and I can write it as the volume fraction of the lipid is not going to change based on the wavelength, but the absorption coefficient will actually change. So, I can write it as like this for a single wavelength, and for a second wavelength I can write it for second wavelength I can write it as mu a lambda 2 f v comma L mu a of lipid for second operating wavelength plus volume fraction of collagen times mu a of collagen for different operating wavelength. Okay. The next question is, what do we know from our knowledge, from our seniors or people who have already researched? What do we already know? We know the absorption of lipid at a particular wavelength lambda 1, for example, right. Not for example, actually we know for most of the wavelengths in the range that we want or the range that we are operating, we know. So, we know the absorption coefficient of lipid at a certain wavelength. We also know absorption coefficient for a second wavelength. We know the absorption coefficient for the collagen for the first wavelength, and similarly we know absorption coefficient for the second wavelength. What do we don't know? We don't know the volume fraction of lipid and volume fraction of the collagen. Okay. So, these are the two unknowns. So, you have unknown 1, you have unknown 2, okay, and you have two equations. Okay. So, what do we do is this is what we are going to get from the experiments. Okay. And we are going to put it inside this equation, and we are going to have two equations. We have two equations, we have two unknowns. So, we can quantify the volume fraction for the lipid and collagen. Make sense? So, this is eventually this is what we are going to do in our next formulation. So, as we have discussed, what do we observe is the effective or the total absorption coefficient is a function of summation of the constituents multiplication of the volume fraction and absorption coefficient. If there are two constituents, then you have two parts. If you have three constituents, you have three parts, and so on and so forth. So, eventually we can write it like this. This is what we have written some time back for two different wavelengths. Okay. And now I need to expand both of them together. Okay. So, using the differential Beer-Lambert law, you can actually simplify this equation as this form over here, and mind it that this mu a is not just mu a, but it is a delta mu a, and this delta mu a means that it is the absorption coefficient at the difference of absorption coefficient at two different wavelengths. Okay. Or it could be a difference of absorption coefficient for two different states at the same wavelength. Okay. As I am saying, without squinching of fist and quenching of fist, right. So, this could be two different states. For example, two different states could be breathing of the person, inhale and exhale, right. This could be two different states, right. So, this mu a is not just a single value, but it is a difference of two different states at that particular wavelength. Okay. Okay. So, here again what do we know? We know this mu a for the wavelength for two different states for lipid and collagen. We also evaluate this optical density. So, again this is a difference of optical density of that particular wavelength, and this is what we evaluate with our experiments. Okay. So, this is what we also know. The unknown is the volume fraction of the lipid and collagen. Okay. So, from these two equations we can actually back calculate the volume fraction. So, this is what we have done, and we come up with these values of volume fraction, and from this we can actually quantify whether it is diseased tissue or it is a normal tissue. Okay. For example, in the case of cancer or tumor, there is more collagen as compared to normal. In the case of normal tissue, there is more lipid content as compared to cancer. So, once we quantify the volume fraction, we can actually get to know if whether it is the part of the tissue is normal or cancer. Okay. So, that is it for this session. The next session we will get to know more about this technique. We will try to learn or we will try to see the actual images of the diffused optical imaging, and we will see which part is normal, which and how we can actually differentiate as cancer. We will talk about the different techniques such as continuous wave diffuse optical tomography, time domain diffuse optical imaging, and then frequency domain diffuse optical imaging. We will also talk about diffusion equation and how we can approximate the diffusion transport equation with simple P1 Pn approximation to simplify the equations and quantify the bulk optical properties. Thank you.